Hi, as Jason just said, my name's Sophie and I received the Rogers Fellowship in Environmental Studies, where I got to do research at the Baruch Institute for Marine and Coastal Sciences in Georgetown, South Carolina. The Baruch Institute was created in 1969 through the combined efforts of the Bell Baruch Foundation and the University of South Carolina. It's an internationally recognized academic research institution, and it has two main missions. The first is obviously to conduct its own original research, and it has numerous scientists that stay at the lab year-round. And the second is they have a huge focus on education, and this means both public education. They have a big discovery center where people can come and learn about projects that are going on, and they also bring classes down from the University of South Carolina to do research there. My project in particular focused on a fish called the mummy chog. It's really abundant in the salt marsh. You can see a picture of it there. And there were three main phases to my project. The first phase was basic population abundance experiments. So what I did was I took these minnow traps, which you can see in the picture, and I went to 4 sites right around the lab, and I just set the traps in, and I went for 3 days and checked the traps every 3 to 4 hours. And I looked for differences in both size and number of mummy chogs caught there. And what I found was that 2 sites consistently produced smaller and less abundant mummy chogs. And I hypothesized that this was due to the isolation of these sites, because the other 2 sites at high tide would flood with the main marsh waters, which meant that they got both an influx of new food sources probably and new mummy chogs, which would lead to the bigger sizes and the higher numbers that I saw at those other 2 sites. The second phase of my project was called geometric morphometrics, which is something I'd actually never dealt with at all until this fellowship. So I'll give you a little basic breakdown of the premise. What you do is you take pictures of your fish and upload them into a computer program and place landmarks on them. And the picture here is the landmarks that I used for my fish. And what the program does is it digitizes these landmarks and gives you coordinates. So what you can do is figure out general fish shape and also things like body length and body depth. And the idea of this is that you take all those measurements as a whole and look at specific variables to see if there are differences. In my case, what I looked at was bottom type. I looked at oyster reef bottoms versus mudflat bottoms. And the reason I thought these might be different is because fish from those 2 types of bottoms have different feeding habits. And I wanted to see if there was different morphology associated with the feeding habits. And where I'm at with this project is I have all my fish collected and all the pictures taken, but I haven't done the statistical analysis yet. I'm kind of waiting on my site mentor to get back to me. So that's where I'm at with stage 2. The final stage I'm actually working on right now in an independent study, and it's population genetics. And what's interesting about the mummy chogs is that even fish that are not very separate geographically can be genetically distinct. So what I did was I collected 40 fish from 4 different sites around the lab, and I'm sequencing the DNA to see if there are any genetic differences basically in those sites. What I learned from this fellowship, I learned a variety of new scientific techniques. I did a lot of ecological surveys, geometric morphometrics, and also microsatellite usage and DNA analysis I'd never done before. I did a lot of statistical analyses with the first part of my project, and I also just learned how to be more independent as a scientist, which is obviously very valuable going on into grad school. The impact of the fellowship, as Dan talked about a lot, you make a lot of great connections through these fellowships. I'm also hoping to get a publication out of the geometric morphometric stuff, so that's really important going into grad school. And it also helped me to determine my educational goals heading forward. Personally, as I said before, I gained a lot of confidence and self-reliance as a scientist and independence. And I was also able to get a lot of advice as I was going through my graduate school applications, which was really valuable. And that being said, I'd like to thank my donor, John Mark Dean, as Jason said, my supervisors, Dennis Allen and Virginia Chervette, and my faculty sponsor, Craig Tepper. Thank you.